Alright, on to the next portion of this video. This portion is in regards to DNA being converted to pre-mRNA, then being converted to mature mRNA. So this process occurs in two specific steps, or non-specific steps, known as transcription, then translation. Within transcription, there are a variety of steps that must be followed for that to occur, and that is essentially just the transcribing of the genes, or the replicating of the genes in RNA, and translation is the translating of those genes, or the conversion of those specific nucleotides into amino acids to produce proteins. Now, here we have an image of a DNA strand. This is a bit more, I guess, broken down or broken into segments, such as the promoter and the RNA encoding sequence. In natural DNA strand, you would just see a bunch of nucleotides together. However, within a segment of DNA, you have the promoter region, and the promoter region is just the region where RNA polymerase recognizes and begins to bind in order to encode the rest of the region, such as here. The rest of the region is known as the RNA encoding sequence, and that contains the 5' untranslated region, the 3' untranslated region, as well as the exons and introns. Now, if we were to expand on this specific model, we see that we have the promoter region. Within the promoter region, we have the TSS, or the transcriptional start site. The transcriptional start site is just the area where RNA polymerase begins transcribing the nucleotides. Then we have the 5' untranslated region, and this is a region that does not code for the gene. However, what it does do is it is coded for because it allows for stability of the mRNA strand. If you'll just give me a moment to find a charger. Okay, we have the 5' untranslated region. That is transcribed. We have the introns and the exons. Those are also transcribed. And within the introns and the extrons, these are the portion of the gene that we are transcribing. Before that, we have the start codon, and the start codon just initiates, or tells RNA polymerase that this gene now begins, and the stop codon is at the end of the gene, and that tells RNA polymerase that the gene has now been coded for. We can stop transcription. And then the 3' untranslated region is just another portion that is also transcribed, and that also aids in stability of the pre-mRNA molecule. Now, for transcription, we have 3 steps. We have initiation, during initiation, what happens is the promoter region is recognized by RNA polymerase, which binds to it, and with the aid of transcription binding proteins, it begins transcription. So, over here in the promoter region, we have a bunch of proteins, and then we have RNA polymerase. These proteins aid RNA polymerase to bind to this specific region, and then once it reaches the transcriptional start site, transcription begins for the entirety of the untranslated region, the gene, as well as the 3' untranslated region. So, initiation, once again, is just when the promoter region is recognized by RNA polymerase and transcription begins. I also want to specify that the transcription binding proteins only help with RNA polymerase binding to the promoter region. They do not help with it transcribing the gene as well as the untranslated regions. It just helps it to begin transcription. The second portion that occurs, or the second step that occurs, is known as elongation. In elongation, RNA polymerase moves along the DNA strand in the 3' to 5' direction, making nucleotides in the 5' to 3' direction. A bit of an image to kind of see how that goes is you'll have the DNA strand and it's going from 3' to 5', and then you'll have RNA. Let's say RNA was in the middle. So right here, what you would have is RNA polymerase adding nucleotides 5' going all the way to 3', making it so that it's anti-parallel. And the third step of transcription is known as termination. And termination is when transcription stops once reaching the termination region, or your transcriptional stop site, which I don't really like to refer to it that much because it's less of a specific stop site similar to the transcriptional start site, and it's more of a transcriptional stop region because a variety of methods are used in order to stop RNA polymerase from transcribing further genes. Once that has all occurred, we then have a premature mRNA. Now, the steps from going from a premature mRNA or pre-mRNA to mature mRNA is a variety of them. So here's kind of an image of the pre-mRNA. So we have the pre-mRNA, we have the 5' untranslated region, we have the introns, the exons, and we have the 3' untranslated region. Now, this pre-mRNA is unable to be exported from the nucleus where it's synthesized into the cytoplasm to where it's going to find a ribosome. It does not have nearly enough protection, and what's going to happen is it might disintegrate on the way out and it just break apart, and then you won't have a gene to be able to translate into a protein. So a variety of steps occur in order to ensure that that can occur. The first step is the 5' capping. So, when 5' capping occurs, you have modified guanosine nucleotides that are added to the 5' position, or the 5' end of the pre-mRNA. And what that does is it aids in the export and acts as protection for the mRNA strand. The second step, or the second thing that occurs, is known as RNA splicing. So you have a specific enzyme known as the spliceosome, and that cuts out all of the introns and the exons, and it removes the introns from the pre-mRNA. The introns are just non-coding regions of the DNA sequence for this gene, and since they're non-coding, they're not needed. So they're removed, and then all the exons are joined together to form one large exon or one large coding portion of the mRNA strand. An easy way to recall that introns are non-coding and exons are encoding is introns are insignificant. Just remember insignificant and introns, and exons encode for DNA or for proteins. E for exons, E for encode. The third step, or the third thing that occurs, is polyadenylation. Now, polyadenylation is just the addition of the poly A tail, which is added to the 3' end of the pre-mRNA, and that stabilizes the pre-mRNA. And the fourth and final step is the export of the mRNA strand from the nucleus to the cytosol, in which it's going to be recognized by a ribosome and translation will occur. So when that occurs, you have transport proteins that transport the mature mRNA molecule into the cytoplasm. And here's an image of the mature mRNA. So the mature mRNA includes a 5' cap and a poly A tail, and it excludes all the introns and instead just has exons. So that's the major occurrences which allow for the pre-mRNA strand to become a mature mRNA strand. It's just the removal of the introns and the addition of the 5' cap and the poly A tail to the 3' end. That is all you need to know in regards to this, or apart from the conceptual aspects. On to Punnett squares and Mendelian genetics. So here we're given an example. We have gray is dominant to ebony, and big G encodes for gray, and little g encodes for ebony. And long wings are dominant to vestigial wings. Big L equals long, little l equals vestigial. And we're going to assume that all the P1 individuals are homozygous. In other words, all of the parental individuals that we're going to cross to one another are true breeding. True breeding means that this individual is homozygous at a specific locus, meaning that whenever it's crossed with another individual who's true breeding or homozygous at that specific locus for any given trait, their offspring will always end up carrying that trait. So if we have two individuals who have or homozygous for an allele that causes individuals to be short, all of their offspring, no matter how many times they mate and that occurs, are going to result in them being short. Doesn't matter how many times you mate them over, doesn't matter how many times you mate their offsprings over, they're always going to end up being short. Unless some strange mutation occurs in which they end up being tall, they're always going to be short. That's what true breeding means. Okay, onto this example. We have a gray individual with long wings, meaning that he's homozygous dominant at both of the loci. So big G, big L, big L. And we're going to cross that individual with an individual who is, sorry, stop it. It's just my cat. We're going to cross that with an individual who is ebony with vestigial wings, meaning that this individual is homozygous recessive at both of the loci. So little g, little L, little L. Okay, now, in order to solve this problem, the easiest way and the easiest method that you can do is by making two separate Punnett squares or making separate Punnett squares for each of the genes. So make a Punnett square for the gene G and make a Punnett square for the gene L and solve for both of those. So for gene G, if we make a Punnett square, put either parent on top or the side, it's your choice. I'm going to put this first parent right here on the top and the second parent on the side. And we see that all of the offspring, or 4 out of 4 of them, are going to carry the genotype big G, little g. Now if we do the same for the L gene, we find that we get the same exact results in which 4 out of 4 of them are heterozygous, or have one big L and one little l. Okay, now what we're going to do is we're going to cross the F1 generation, or this generation. In order to find the genotypes of the F1, we simply take GG, or 4 out of 4 of the GGs, big G, little g, multiplying that with the 4 out of 4 for the big L, little l, and we find that we get 16 out of 16 of the individuals having the genotype big G, big L, little l, or 100% of them. So we know that the genotype for the F1 is big G, big L, little l. Now we're going to cross two individuals from the F1 progeny. Now if we were to cross them, we would get big G, big L, little l crossed with big G, big L, little l, and we want to follow the same steps we did for the previous problem, in which we're going to separate the genes and then perform a Punnett square for both. For gene G, we have a big G and a little g, a big G and a little g, we get a quarter of them having the genotype big G, 2/4 of them, or 1/2 of them having big G, and 1/4 of them having little g. And it's preferred to keep this in the 2 out of 4 ratio, just because you will see why as we continue this problem. Now for the L gene, if we do the same, we have big L, little l, big L, little l, big L, big L, big L, little l, big L, little l, little l, little l, and we'll end up with the same ratios of a quarter of them being homozygous dominant, half of them being heterozygous, and a quarter of them being homozygous recessive. Now, once we're here, we can find the phenotype ratios of all the individuals. So for an individual to be gray with long wings, the genotype has to be at least one big G. The other allele doesn't matter, so I'm going to put this little underlined to signify that any other allele could be present. We know that since this is following the typical dominant recessive pattern, that one dominant allele is enough to express the dominant trait. Likewise, for the long wings, we have to have at least one big L. The other allele doesn't matter. So, if we look at our punnet square, we have 1 out of 4 of them that are homozygous dominant. So we have 2 alleles, and that will result in the individual being gray. And we have 2 out of 4 of them who are heterozygous who have at least one big G allele. So we can combine these ratios together to get a 3 out of 4 ratio. So 3 out of 4 of the individuals of this progeny will be gray because they have at least one dominant allele. So we can put that ratio down, 3 out of 4. And we're going to multiply it with the ratio of having long wings. To have long wings, you have to have at least one big L. Here we have 2 big L's, and here we have 1 big L. Combine the ratios together, we have 3 out of 4. So 3 out of 4 times 3 out of 4 equals 9 out of 16. To have gray coloring with vestigial wings, you have to have one big G, the other allele doesn't matter. And to have vestigial wings, you have to be homozygous recessive, meaning you have to have 2 little L alleles. So we know that the ratio for having a gray color is 3 out of 4. Multiply that with the ratio of having 2 little L's, or being homozygous recessive for the L gene, is 1 out of 4. So 3 out of 4 times 1 out of 4, that equals 3 out of 16. To have the ebony color with long wings, you have to have 2 little G's with a big L, and the other allele doesn't matter. So we're going to multiply the ratio of having 2 little G's, or being homozygous recessive at the G locus, and that is 1 out of 4, as we can see over here. So 1 out of 4 times the ratio of having long wings, which is 3 out of 4, times 3 out of 4, and that equals 3 out of 16. In order to have ebony skin color, in order to have the ebony phenotype, you have to have 2 little G's. In order to have vestigial wings, you have to have 2 little L's. The ratios for both of those are 1 out of 4, so 1 out of 4 times 1 out of 4. That will result in 1 out of 16. Now, if we look at the results that we got, we have 9, which are going to be gray with long wings, 3, which are going to be gray with vestigial wings, 3, which are going to be ebony with long wings, and 1, which is going to be ebony with vestigial wings. Now, this is significant because this follows Mendel's law of independent assortment. That means that these genes, whenever you cross 2 individuals who are heterozygous for a gene, you're going to get this specific ratio for all the different genotypes. And that is because the genes, they are located on different chromosomes and they assort independently. If we were to perform a cross and we find that the ratios that we get are different from what we predicted, that would infer that the genes are located on the same chromosome, or there is an issue with assortment. So this is the typical Mendelian ratio which will occur, and this is what's normal. And this method I like to use because it helps you in finding the specific genotypes that you're after as well. If we were asked to find the ratio of an individual who's going to have the genotype big G, little l, we simply take the ratio for big G, which we know is 1 out of 4, multiply it by the ratio of having little l, which is 1 out of 4, and we get 1 out of 16. So this tells us the ratio of each of the genotypes we can get. And you can go and solve for all of them, and you will find that they'll all add up to 16 out of 16. So that's how you know that you did the problem right. Okay, on to the next portion of this, which is Mendelian genetics and biochemical pathways. So in this example, we have a pathway for producing pigment. Here we have the colorless, and that acts as the precursor molecule. And colorless is converted by enzyme A into the blue color, and enzyme B converts the blue color into orange, and enzyme C converts orange into green, which acts as the final product, and that's the final pigment that is produced, and that's what is expressed. In order to convert to the final color, you have to have orange. If you are unable to convert orange or blue into orange because there was an issue with the enzyme B, then you would express the blue color. Likewise, if there was an issue in converting the colorless color to blue because enzyme A was defected, then you would result in the organism showcasing a colorless color. And we can apply this to Mendelian genetics. So, if we were to have a mutation at any of the alleles, and we have two individuals with the same mutation who mate, and then they end up having an offspring who carries both of the alleles with the mutations, then that color is going to be, then the color cannot be converted and expressed. So, for whatever the precursor to that mutation was, that will be expressed. So if the precursor to the mutation of enzyme B is blue, and we were to inherit two of these alleles with this same mutation, then the color that the individual or the offspring would have is blue. And here's a smaller example. If we have white and that's converted by enzyme A into yellow, and that's converted by enzyme B into black, if we have little a, which is present, and that means that enzyme A is mutated, and we were to have two of those same alleles with the same mutation, then they would not have the proper enzyme to convert the white color into yellow, so therefore, the expression of that individual would be white. Likewise, if we were to have two mutated alleles of enzyme B, then yellow would be expressed since they cannot produce an enzyme that will convert yellow to black. So here's an example that we're given. We're crossing an individual who's green with an individual who is yellow. And since we know that these genes are on different chromosomes, they will assort independently, and so we can make a Punnett square for each of the genes. So if you make a Punnett square for gene A, I want to use the black color, gene B, and gene C. If we make a Punnett square for gene A, we see that both of them are homozygous dominant, so they're both going to be big A, big A, big A, big A. They will result in all of the offspring having the homozygous dominant genotype. So 4 out of 4, or 100% of them will have AA, big A, big A. For the B gene, we see that this yellow individual is homozygous recessive, and this green individual is homozygous dominant. So big B, little b, crossing them together will result in all of them being heterozygous. So 4 out of 4, or 100% of them, are going to have the genotype big B, little b. For gene C, we know that they are all homozygous dominant. So all of the offspring are going to carry the homozygous dominant genotype. So 4 out of 4 of them will be big C, big C. Now, we know the genotypes are simply going to be big A, big B, little b, big C, big C. Now what we're going to do is we're going to cross the F2 genotypes, or the F1 genotypes, to produce the F2 genotypes. So we're going to cross this individual with another individual from the progeny, which is going to have the same genotype. So cross that with big A, big B, little b, big C, and we're going to perform the same exact thing we did for this portion 1 of the problem, in which we're going to make Punnett squares for each of the genes, and then we're going to solve them out. So for A, we know that 4 out of 4 of them, since they're both still homozygous dominant at that gene, are going to be big A. For C, we know the exact same thing. 4 out of 4 of them are going to be big C, since they're both homozygous dominant. Now for B, we know that both of the organisms are heterozygous. So if we were to make a Punnett square for them both, we would get 1/4 of them being homozygous dominant, 1/4 of them being homozygous recessive, and half of them being heterozygous. Now, we want to find the amount of the individuals who will be expressing the green color and who will be expressing the yellow color, because we know that any individual who has little b is going to express the yellow color, since if we look at the pathway, enzyme B is responsible, okay, this blue should be yellow, it should not be blue. Okay, so if we look at the pathway, we see that enzyme B is responsible for converting the yellow color into the orange color, which is then converted by enzyme C into the green color, or the final color. So if we were to have mutations at enzyme B and we were to have two alleles that have the same mutation, which are in an individual, then that individual would be unable to convert the yellow pigment into the orange pigment. If we have one of the mutations, that's fine, because one of the alleles is going to allow for the production of that specific enzyme to convert that pigment from yellow to orange, and that's enough to be able to express the green color. So in order for an individual to be green, they have to have the genotype one big A, the other allele doesn't matter, one big B, the other allele doesn't matter, and one big C, the other allele doesn't matter. In order to have the yellow genotype or yellow phenotype, you have to have one big A, the other allele doesn't matter, two little b's, and the c's don't matter. They could be, this is all we need. Everything else doesn't matter. So, if we're looking at the green genotype, we know that the ratio for having the big A allele is 4 out of 4. We're going to put that down. So 4 out of 4 times the big B. We have 1/4 that are homozygous dominant, and 2 1/2, which is heterozygous, meaning they contain at least one big B allele. We can combine these together to get 3 out of 4 ratio, times 3 out of 4, times the ratio for at least one big C, which we know is 4 out of 4. So if we multiply that together, we get 16 times 3, which is, if I'm not mistaken, 48 over 64. And the ratio to have the yellow coloration is one big A, at least we know is 4 out of 4, times two little b's, which we know is 1 out of 4. And for the other allele, since it doesn't matter, if it was big C, little c, or little c, big C, none of them matter. We can combine all of those ratios together. So if this was, let's say, 1 out of 4, 2 out of 4, 1 out of 4, we can combine these together to get 4 out of 4, and we would use that. So times 4 out of 4, which gives us 16 out of 64. To check to see if my math is correct, I'll just add them together, see if they equal 48 and 16. That equals 64, so my math is